This is Biology 1, Chapter 12, Part 4 on molecular genetics. In this portion of the video lecture, we will be discussing the three types of RNA and discussing the process of transcription, which is when we make RNA from DNA. In order for a cell to understand what the genes are coding for, or for the cell to read what information is contained in the genes, there is a central dogma or a central idea of how this process works. And that process is to first make DNA, then make RNA, and then make proteins. So the information from the DNA is coded into RNA, which is then translated into proteins. And remember, your cells, that's basically their number one job, is to make proteins. There are three forms of ribonucleic acid, or RNA, and each form has a particular shape and a function or job associated with it. The most common type of RNA in your cell is called messenger RNA, which we will abbreviate with a little m, RNA. It is an uncoiled single-stranded molecule. It is not a double helix like DNA. The job of the messenger RNA is to be a messenger. Messenger RNA transmits information from the DNA to make proteins. And just like a bike messenger, the messenger RNA travels from the nucleus, which is where all the DNA is, to the ribosome with that information. And remember, the ribosomes are the protein makers of the cell. So if you need to get your information about what proteins that are to be made, you're going to need to get that and travel from the nucleus to the ribosome. So messenger RNA, that's the job of mRNA, is to transmit that information from nucleus to ribosome, so carrying the message. The ribosome itself has its own particular special RNA. We call it ribosomal RNA, or little r RNA. The shape of ribosomal RNA is globular, for lack of a better word. There's no particular shape. It's ribosomal shaped. The function of rRNA is the actual site of protein assembly. So it's the ribosome itself. Transfer RNA is the third type of RNA. We label that with a little t, RNA. To describe the shape of the transfer RNA molecule, it kind of is like a funky hairpin. There's 23 different varieties of transfer RNA. And the job of transfer RNA is to bond to a specific amino acid. Remember, amino acids are the building blocks of proteins. So tRNA bonds to an amino acid and then translates that information to be made into proteins. The process of making RNA is called transcription. When you transcribe something, you're writing it down. So making RNA from DNA is very similar to, for example, writing in pencil and then writing the same thing in ink pen. It's pretty similar, but it's got that one little difference of pencil to ink. So it's a similar process to how we get into the DNA molecule, because we're making the RNA from the information on the DNA. So if we need to get into our DNA molecule, we need the help of the enzyme DNA helicase. Remember, helicase unwinds and unzips the DNA molecule. It breaks those hydrogen bonds so we can get the two strands apart. Now remember, there's a leading and a lagging strand on DNA. Because messenger RNA is only one strand, it makes the most sense to use the easiest strand to get the information to make RNA. Because the leading and the lagging strand on DNA are complementary, we really don't need to use both strands to make a single-stranded molecule. So only the leading strand of DNA is used to make RNA. The lagging strand just kind of hangs out and waits to be sealed back up by DNA ligase. So after DNA helicase unwinds and unzips the helix, an enzyme called RNA polymerase will add RNA bases. So every time an RNA sees a guanine on the other side of the DNA molecule, it's going to put down a cytosine. If it sees a thymine, it's going to put an adenine. And here's where the difference comes in. Remember, uracils are unique to messenger RNA. So if RNA polymerase sees an adenine on the other side of the DNA molecule, it's going to put a uracil across from that. So A pairs with U and C pairs with G for RNA. Now, in order to make sure that the cell's mistakes, if there has been any that hasn't been caught up to this point, if there are no mistakes made, then in general what happens is every single base on the DNA is not used to make RNA. Again, the cell makes mistakes. In interphase, if during the S phase of the replication of DNA, if there were some accidental switching of base pairs, then in the final RNA processing, that information won't be used. There are two types of areas on the DNA molecule. The introns are what we call intervening sequences. They are not coded for. Introns are commonly called junk DNA, although really there is no DNA that is junk. You do need this DNA because it does need to be cut out, which sounds strange. But again, if a mistake was made in the DNA copying process and that mistake is in an intron, it's not going to make any difference to the organism because those are literally cut out. Special editing molecules called SNRPs come along and cut out the introns. The remaining pieces of DNA that are left are called exons. These are the expressed pieces. These are the pieces of DNA that are coded for in the final protein making process. So the exons hold the actual genetic information that needs to be read and coded. If there's a mistake in the exon, there may be some functional problems with the organism. If a mistake is made in the intron, it doesn't matter because those are cut out and the bases that were used for that are kind of recycled back into the nucleus to be used again. So this is one of those mistake making processes that will hopefully reduce the amount of mistakes that could be made during the DNA replication process. Your cell has lots of backup plans and lots of checks and double checks and triple checks to make sure that the DNA is coded for correctly and that the RNA is transcribed correctly as well. Again, everything's got to go 100% correct 100% of the time. The process of transcription also needs to do that as well. Introns at the end need to be cut out. Exons need to be glued together. This has been part 4 of chapter 11 or 12 rather on molecular genetics.